Fine control of sound frequency tuning and frequency discrimination acuity by synaptic zinc signaling in the mouse auditory cortex. Presentation by Carol Ann Bailey. The brain is constantly encoding sensory information that will direct behavior. Cortical neurons within the brain react to sensory stimuli and are tuned to specific sound frequencies. This tuning is directed by thalamocortical input and the interaction of excitatory and inhibitory intracortical inputs, where cortical inhibition regulates stimulus selectivity. However, the underlying mechanisms that regulate cortical distinction to sound frequencies is unknown. Our objective is to explore how synaptic zinc plays a role in cortical frequency tuning and the regulation of sound frequency discrimination acuity within the auditory cortex of a mouse's brain. The brain contains cortical neurons that respond to different sound frequencies. In the auditory cortex, synaptic zinc was discovered to play a role in regulating sound processing. Synaptic zinc is released from different brain areas, including the amygdala, hippocampus, and sensory neocortex. The brain releases synaptic zinc from glutamatergic vesicles to regulate neurotransmission by fine-tuning AMPA receptor and NMDA receptor excitatory postsynaptic currents. Since we are aware that synaptic zinc has a role in sound processing, we are investigating the role of synaptic zinc and its influence on cortical frequency tuning and its modulation on sound frequency discrimination acuity. In order to conduct this experiment, we used 5 groups of mice, including ICR mice, ZNT3 knockout mice, PV-CRE mice, SOM-CRE mice, and ZNT3-WT mice. We prepared each mouse for in vivo imaging by injecting them with AAV and waiting 11 to 24 days. Part of the experimental setup was a speaker that was placed 10 centimeters away from the rodent's left ear and was set to play low frequencies. We utilized fluorescence imaging and 2-photon imaging. We also placed electrodes in the subdermal of the mouse's brain to record responses. To analyze all of our data, we used MATLAB and QuickCalcs. We began the experimental procedure by locating the primary auditory cortex. We did this by utilizing in vivo transcranial fluorescent imaging. We used AAV, which is a calcium indicator of GCAMP6F fluorescence, in order to see which area of the brain lit up when a low-frequency sound was played. Due to the mirror-like reversal of tonotopic gradients between A1 and AAF regions, the low sounds activated particular low-frequency regions in the A1 and AAF regions of the auditory cortex. Next, we performed a craniotomy in order to do 2-photon imaging to view the effects of synaptic zinc on layer 2-3 of principal neurons. This figure, figure 2.1, shows various aspects of the experimental procedure. Part A shows the setup of the transcranial imaging. Part B shows the fluorescence imaging. Part C shows the craniotomy, and Part D shows the 2-photon imaging utilizing fluorescent imaging. In order to explore the role of zinc and frequency tuning, we injected the mice with an extracellular zinc-specific chelator, ZX1, and viewed changes in the sound-evoked response by the in vivo fluorescent imaging. We explored the relationship of ZX1 with the 5 different groups of mice. We looked at the ICR mice, which are the control mice, the PV-CRE mice, which have an inhibition to principal neurons, the ZNT3 knockout mice, which are the mice that lack synaptic zinc, the SOM-CRE mice, which have an inhibition to both principal neurons and PV neurons, and the ZNT3-WT mice. We then compared the bandwidth of layer 2-3 principal neurons and created a heat map in order to see differences in response between the 5 groups of mice. We also performed a test to see if disruptions in zinc signaling would affect the mouse's ability to distinguish varying sound frequencies. We did this by using a pre-pulse inhibition to acoustic startle reaction experiment between the ZNT3 knockout mice and the ZNT3WT mice. Figure 2.2 shows the effects of ZX1. Graphs E through F show a comparison of ZX1 and a control on principal neurons. Graph H shows a heat map of the changes that occurred after ZX1 was injected. Overall, this data shows that synaptic sync increases the frequency tuning of A1 layer 2-3 principal neurons. This figure, figure 2.3, illustrates the results of the pre-pulse inhibition of the acoustic startle reaction. This behavioral task was conducted in order to see if disturbances in synaptic zinc signaling would diminish the mouse's ability to distinguish sound frequencies. These results show that ZNT3 knockout mice have decreased abilities to distinguish between frequencies. Results of this study revealed that ZX1 widens the bandwidth of the receptive field of layer 2-3 principal neurons to sounds of 30 and 70 decibels. ZX1 also increased the bandwidth and increased the number of sound frequencies for A1 layer 2-3 PV neurons at 70 decibels. This shows that zinc sharpens frequency tuning in these neurons in the auditory cortex. Consistent with previous studies, it was evident that there is a relationship between ZX1 and the BF area. When a sound was near BF, ZX1 showed significant decrease in response amplitudes. When a sound was away from the BF region, ZX1 showed an increase in response amplitudes. WT mice lost the capacity to distinguish between sounds with small frequency changes, such as a change from 16 kHz to 15.2 kHz. This loss of the ability to distinguish differences in frequency was due to an injection of ZX1. Based on the results of this study, synaptic zinc enhances the amount of responses to sound frequencies near BF and increases the A1 layer 2-3 principal neurons' receptive fields by decreasing responses to sound frequencies away from the BF. This shows that synaptic zinc is a unique cortical signaling pathway that is used within the auditory cortex and aids in frequency discrimination and frequency tuning. There are 3 proposed models that strive to explain the tuning of auditory neurons. These models are the iceberg model, the approximately balanced model, and the lateral inhibition model. These models are not exclusive and work together. Based on this study, synaptic zinc plays a role in each of these and is a link between further understanding of how inhibition impacts cortical frequency tuning. Cortical mechanisms are important for sensory processing of the world and are necessary for stimulus selectivity. Within this study, we discovered that synaptic zinc plays an important role in auditory processing and the regulation of cortical responses. Synaptic zinc both enhances auditory acuity and shapes auditory processing. Without synaptic zinc, there are deficits in the auditory cortex of the mouse's brain. These deficits are similar to deficits found in individuals with schizophrenia and dementia. There are various ways that we could improve this study and further explore this topic. We could enhance the levels of zinc at specific synapses for zinc transporter balance to further investigate zinc with frequency tuning and discrimination. In this study, we had infrequent sound frequencies. So, for a further study, we could increase the sampling density of sound frequencies in order to have more in-depth measurements, particularly for regions farther from the BF. We could also dig deeper into ZX1 and its role in spontaneous activity. We could further delve into the depths of tonic zinc and zinc with regards to spontaneous activity in the primary auditory cortex. And another area of further research that we could delve into is the connection between synaptic zinc, auditory processing deficits, and deficits in those with schizophrenia.